Every human being carries a ghost inside them, fragments of DNA from ancient populations that vanished tens of thousands of years ago. Most of these genetic echoes are small, scattered across the genome. But in the Americas, scientists found something different. A gene inherited from an extinct human species appears in millions of people today, even though that species never set foot in the Americas. So how did a Denisovan gene become a survival tool for the first Americans? The answer rewrites how the Americas were populated. For most of the 20th century, the story seemed settled. Around 13,000 years ago, hunters crossed the land bridge from Siberia into Alaska. They carried distinctive stone tools. They were the Clovis people, and they were the first. Clean, simple, textbook. But whispers of contradiction were already surfacing. Sites across both continents kept testing older than they should. Monte Verde, Meadowcroft Rock Shelter. Human presence pushing back further in time, refusing to stay inside the neat boundaries researchers had drawn. Something was wrong with the simple story. In the early 2000s, geneticists analyzed mitochondrial DNA from living indigenous Americans. They expected to confirm the Clovis timeline. Instead, they found 5 founding lineages, all tracing to a single ancestral population that crossed from Siberia. The timing pushed back, not 13,000 years, but closer to 20,000. Then came the real disruption. In 2010, a team led by Svante Pääbo sequenced something unexpected from Denisova Cave. DNA that didn't match anything in the record. It belonged to an entirely new kind of human. They called them Denisovans, a population that split from Neanderthals hundreds of thousands of years earlier, occupying vast stretches of Asia before disappearing. When researchers scanned living populations, they found Denisovan DNA scattered across the globe. In Melanesia, up to 5%. In Tibet, a Denisovan gene helps people survive extreme altitude. In Aboriginal Australians, in Filipinos, in people across Asia and the Pacific. But the Americas remained blank. The assumption held. The first Americans had no Denisovan ancestry. That assumption was about to crack. In 2017, a team scanning genomes flagged a region on chromosome 12 in Mexican populations. It kept appearing as an outlier. Signals of positive selection, high frequencies of something unusual. The statistical signatures were clear enough to notice, but ambiguous enough to dismiss. Was it Neanderthal? Was it something else? The standard models couldn't explain it. Some argued it was just noise, statistical artifacts. Others suspected complex population structure in Asia before the crossing. The debates remained technical, dense, confined to journals. The region in question, a gene called MUC19, seemed almost mundane. MUC19 produces mucins, the proteins that make mucus. But mucus is the body's first line of defense, the barrier between you and every pathogen you breathe. A gene that shapes mucus isn't trivial, it's survival. And inside this gene, something was hiding. In 2023, Fernando Villanea at the University of Colorado Boulder and his colleagues decided to stop ignoring the anomaly. Their question was simple. What exactly is going on at MUC19 in American populations? They analyzed genetic data from Mexican ancestry individuals. What they found stopped them cold. Within MUC19, the sequences split into 2 distinct groups. One matched standard human variants. The other matched the Denisovan genome almost perfectly. Not similar, nearly identical. The Denisovan-like sequence appeared at high frequency in indigenous American populations. In ancient individuals who lived before European contact, it was even higher. This was not recent mixing. This was deep ancestry, preserved across millennia, hidden in plain sight. But here was the impossible question. How did Denisovan DNA reach the Americas when Denisovans never crossed Beringia? of how Villanueva's team checked the Neanderthal genomes. That's when the story twisted. The Altai Neanderthal from Denisova cave showed no trace of the MUC19 variant, but two other Neanderthals, one from Chagirskaya cave and another from Beringia cave, carried it. Both had the Denisovan version. This changed everything. The variant hadn't jumped directly from Denisovans to modern humans. That path was blocked. Instead, it moved first from Denisovans into Neanderthals somewhere in Siberia tens of thousands of years ago. Those Neanderthals later encountered modern humans migrating out of Africa, and they passed the variant on. Denisovan DNA inside a Neanderthal genome, inherited by modern humans who would eventually cross into the Americas. Now, picture one person, an ancient individual buried in the Americas more than 10,000 years ago. Their genome carries this Denisovan Neanderthal variant at MUC19. They have no name for it, no knowledge of where it came from. But when they breathe the cold, dry air of an Ice Age landscape, when their body encounters pathogens never before seen by human immune systems, this gene is working, producing mucins, shaping the protective layers lining their respiratory tract and digestive system, a shield they never asked for from donors they never met. This person survives, their community survives, and the variant spreads. When researchers surveyed modern populations, the distribution told a story. About 1 in 3 people with Mexican indigenous ancestry carry the Denisovan-like MUC19 variant. In Peru, about 1 in 5. In Puerto Rico and Colombia, it drops to around 1%, tracking directly with indigenous ancestry levels. In Europeans, the variant is almost absent. In Africans, 0. But there was another layer. Genes often contain repeating sections, like a stutter in the code. More repeats mean more protein produced. Think of it as a dimmer switch. More repeats, brighter protection. Indigenous American populations carrying the Denisovan variant showed massively expanded repeats, hundreds of them, far more than any other population on Earth. Something had happened after the variant arrived. Natural selection hadn't just preserved it. Selection had amplified it, pushed it to extremes. The leading hypothesis is immunity. The logic is brutal. The first Americans entered a continent filled with unfamiliar pathogens, new viruses, new bacteria, new parasites. Their immune systems, shaped in Siberia for thousands of generations, suddenly faced threats they had never encountered, threats their bodies didn't recognize. Mucus is the first barrier. It traps pathogens before they reach vulnerable tissues. It contains antimicrobial compounds. It flushes invaders away. A gene that strengthens this defense would be powerfully favored. The Denisovan variant didn't just tweak MUC19. It provided a template for expansion. Selection didn't just preserve the variant, it multiplied its effect. Evolution responding to pressure. Fast, decisive, effective. So here is the answer to the opening question. How did a gene from a ghost population become a survival tool for the first Americans? It moved from Denisovans into Neanderthals in ancient Siberia. It was carried by Neanderthals who later mixed with modern humans somewhere in Asia. And it was inherited by a population that would eventually cross Beringia into a new continent. In the Americas, that gift met its moment. Confronted with novel pathogens, selection seized the variant, amplified it, and spread it through generations. A genetic inheritance buried for millennia, waiting, silent, then essential. The old story was simple. Modern humans left Africa, replaced all other populations, and populated the world as a single lineage. Winners remained, losers vanished. But the evidence now shows something different. We are composites, mixtures, hybrids, products of encounters across deep time. The Denisovans didn't disappear completely. They persist in our immune systems, in our ability to survive at high altitude, in our responses to pathogens. They persist in mucus genes carried by millions today. The populations that crossed into America were not representatives of a single ancestral group. They carried fragments of Neanderthals, fragments of Denisovans, fragments of others we have not yet discovered.
What does this tell us about identity? Ancestry is not a line, it is a web. Survival is borrowed from those who came before, even those we would not recognize as human. The past does not stay in the past. It travels inside us, encoded in molecules that shape our bodies every day. As Fernando Villanea explained, indigenous American populations expanded from a small ancestral group near the Bering Strait to occupy every biome in the Western Hemisphere. Deserts, rainforests, mountains, tundra. They did it in a geological instant, and they did it with help from genes that had already faced other worlds. The fragment from Denisovan cave is small. It is all that remains of a young woman who lived 75,000 years ago in the Altai Mountains. Her population is extinct. Her face is unknown. Her language, her songs, her stories, gone. But her genes are still here, carried by people who crossed ice and tundra, who spread through deserts and rainforests, who adapted, survived, and passed on what they carried. You carry your own hidden ancestors, threads stretching across continents you've never seen, across species you've never named, encoded in your DNA, waiting. Subscribe to GenealogyX because the past is not what you thought, and it is closer than you think.